Right, today we are going to talk about Rh blood group system. But while discussing Rh blood group system, I would love to compare and contrast the Rh blood group system with ABO blood group system, right? As we know that these blood group systems consist of antigens and antibodies, right? Now, first of all, we'll come to that Rh blood group system, how it was discovered. Rh blood group system, how it was discovered and why it is named as Rh. You must be knowing Rh basically stands for rhesus monkey. Rhesus monkey. What really happened that around 1940, right, during some experiments, red blood cells from the rhesus monkey, that's a happy monkey, red blood cells from the rhesus monkey were injected in some small animals like rabbit, RBCs. From the rhesus monkey, they were transfused, these red blood cells, into the circulation of rabbit or guinea pig, right? And then they found that the small animal was making antibodies, right? And those antibodies were able to cross-react or agglutinate 100% of the red blood cells from all rhesus monkeys. Again, listen. We took red blood cell from this monkey, injected into rabbit, right? Of course, this red blood cell which is brought from monkey to the rabbit, this red blood cell has many antigens, right? And many of these antigens are foreign to the rabbit system, right? Now, rabbit will make antisera, right? And these antisera were tested on RBCs derived from rhesus monkeys, and all rhesus monkeys, for example, you chose 100 monkeys, and in all those monkeys, their red blood cells were able to react with this antisera. Is that right? So it was speculated that red blood cells of the monkey contained an antigen, and that antigen was named as rhesus antigen. That was named as rhesus antigen. Later on, the same antibodies were tested on human RBCs. They were tested on human RBCs, and it was found that the same antisera was able to, able to agglutinate or react with red blood cells from almost 85% of white population. Right? Again, I will repeat. What was happening? That whatever antisera was produced by the rabbit against the RBC's antigens of rhesus monkey, same antisera was able to react with the red blood cells or agglutinate the red blood cells from almost 85% of white population, Caucasians, right? European population, white European population. So, but in about 15%, in 15% of the population, red blood cell did not react with this antisera. Is that right? Now, what does it mean? It means simply that when rhesus monkeys RBCs were entered into rabbit or guinea pig, the antisera which was derived, of course it was able to agglutinate red blood cells 100% derived from all the rhesus monkeys, but surprisingly it was also able to agglutinate the red blood cells derived from white population of about 85%. right? But it was not able to, anti-sera was not able to agglutinate or react with 15% of the white population. Then it was speculated that this 85% population, right? This 85% of white Americans or Westerners population, right? They are having something common in their red blood cells and monkey red blood cell, right? That this antigen which is present on this rhesus monkey, it must be present over here too. And this antigen should not be present over here. Am I clear to everyone? It's so simple. Now, all those people whose red blood cells were agglutinated or reacting to this anti-sera, they used to say they are having antigen, yes, antigen similar to rhesus monkey RBC, or they simply call it, these people are having Rh antigen. What they were calling? Rh or rhesus antigen. Or they simply said these are Rh positive people. These people are Rh positive. And here those people who are not reacting to that anti-sera, right, they said that these are Rh negative. Before I really go ahead, I should be thankful to the doctors, our people who discovered all this thing. They just call it Rh positive. Thanks God they didn't call it monkey positive. Otherwise many of us will be monkey positive and some of us will be monkey negative. Right? So thanks God, when we are talking as doctors, we say I'm Rh positive. And we don't say we are Rh monkey positive. Right? So let's go back. So this is initially how this Rh system was discovered. Right? Now I will go into detail. Up to now we have just seen that basically those people who have Rh antigens on their red blood cells, they are considered Rh positive, and those people who do not have Rh antigen on their blood, they are considered Rh negative. But things are not so simple. Let's go into detail. Because this story was in 1940, but now so many years have passed, decades have passed, and we have learned so much about Rh system. So now we remember that now Rh system consists of not one antigen. It consists of a whole system of antigens. Up to now there are more than 50 Rh antigens. There are more than 50 Rh antigens, but some of them are more important and others are less important clinically. Right? So let's suppose here is an RBC. Right? Now, this is red blood cell from an Rh positive person. From an Rh positive person. It means this red blood cell has Rh antigen. Actually, what is exactly the nature of Rh antigen? What it is? What is its structure? Because it's important to know, because if we know the nature of the Rh antigen, we can speculate what kind of antibodies will be formed against those antigens. Basically, Rh antigens are proteins. Rh antigens are proteins, and these are the protein peptide chain which passes through this is a peptide chain, and this peptide chain passes through the red blood cell membrane 12 times. How many times? 12 times. That is why we call it 12 pass membrane proteins. Membrane proteins. And these proteins are the integral part of red blood cell membrane. They are the integral part of red blood cell membrane. That is why we also call them membrane proteins. So Rh antigens are fundamentally proteins which are embedded within the membranes of red blood cells, and they are 12 times they passes through the membrane. So they have 1, 2, 3, 4, 5, 6 extracellular loops and 6 intracellular loops. And these are, of course, if this is protein or peptide chain, one end should be amino end and other should be carboxyl end, and both of amino and carboxyl end are intracellular, right? Antigenic determinants are over these loops. Antigenic determinants or epitopes are on the extracellular loop. Is that right? Now, later on it was discovered that Rh antigens are of different types, right? Most common and very strongly immunogenic is D antigen. Let me put it over here. This is the D antigen, right? Now what exactly it is? D antigen, this is one epitope of the D antigen I'm showing. This D antigen is part of this peptide chain. So it means it is a special sequence of amino acids, right? So we can say D antigen is basically one of the, yes, component of or amino acid sequence of this 12 pass membrane protein. Now, as I told you that Rh antigens, there are more than 50 discovered. Out of them, the 5 are very important. Five antigens are very important. One is D, other is capital C or at its place small c, or capital E or small e. So how many are there? Capital D antigen, capital C antigen or small c antigen, capital E antigen or small e antigen. Remember, there is no small d antigen. When we say there is small d, actually we mean there is no d, right? So these Rh antigens are also called Rh factors. These Rh antigens are also called Rh factors. So out of this, more than 50 antigens or more than 50 Rh factors, the most important are D, C and C, E and small e. All of them are slightly different from each other. Now here I want to highlight one more thing. Now this is a peptide chain which is having D antigen. Actually D antigen has many, around 4 antigenic determinants, but I'm just showing one, right? Then what about C and E? There is another. This is another molecule, and on extracellular loop number 2, there can be either capital E or C or small c, right? Remember D I have shown with these things. Why I'm showing like this? Because it is very strongly immunogenic. Once D antigen enter in the body of a person who is not having D antigen as self antigen, this may induce a strong immunological reaction. Then this is C, and on 4th extracellular loop there is another here, and this is E. So what I have shown, okay, let me make the color different. This is D, right? Here it is C, and here it is E. Right? Now, basically, these proteins are coded by the genes. They are coded by the genes. But RBCs don't have genes. Do RBCs have genes? They have genetic material? Nucleus? So from where these genes, how these membranes can make such proteins? Yes? Erythroblast, yes. Erythroblast and precursor cells of the red blood cells, they do have genes. And there are two genes which are responsible to make these antigens. One gene is, of course, they are not in RBCs, they are in precursor cells of the RBCs like proerythroblast. One gene is, I can say, Rh-D gene, which is making this molecule. And other is Rh-CE, which is making this molecule. Later on we'll discuss about these two genes, right? For a while you just trust me, then there are, as far as these important variants of Rh factors or antigens are concerned, Rh-D are by the Rh-D gene, Rh-D factor, and Rh-C and E, they are determined by presence and function of the Rh-CE genes. Is that clear? Now, these are Rh antigens. Let's compare them with ABO system antigens. Let's compare them with ABO system antigens. ABO system antigens are not proteins. ABO system antigens are not proteins. They are basically carbohydrates. They are small chains of monosaccharides. Those small chains of monosaccharides are called oligosaccharides. So basically, blood group A, it has one type of chain, and blood group B has different type of chain. Now, people who have blood group A, they are having A antigens on their red blood cells. A antigens are basically oligosaccharides. People who are blood group B, they are having B antigens, and B antigens are also oligosaccharides. And people who have blood group O, they do not have A and they do not have B antigens. Am I clear? Now, if someone asked what is the major difference what is the major difference in the antigens of Rh system and ABO system, the first answer should be Rh system antigens are proteins. Rh system antigens are proteins, and ABO system antigens are oligosaccharides, carbohydrates. An easy and funny way to remember that we can say Rh antigens are like chicken pieces, and these are the sugar candies. These are the sugar candies. So if your RBCs, again next time if you are confused, just remember, people who are Rh positive, they are having proteins, antigens or chicken pieces, and people who are ABO, right, ABO group, they are basically having sugar candies, carbohydrates, oligosaccharides. Am I clear? Any question up to this? Right. Now, we move forward. Out of these, I told you there are more than 50. Out of those 50, 5 are the most important. Either big D antigen is important, big C or small c, big E or small e. But among all of them, the most important is D antigen. Most important is D antigen or Rh-D factor. We call it Rh-D antigen or factor. Now this Rh-D, how many types? Rh-D, Rh, yes, C, and then Rh, small c, right? Then Rh, big E, and then Rh, small e. Right? So these are 5 most important Rh antigens. But among them, which one is the most important clinically? Most important is Rh? D. Why it is so important? Because number one, it is most prevalent. It is most prevalent, most commonly found. And number two, it is most immunogenic. Most immunogenic. Most immunogenic mean if a person who does not have D antigen, a person who on his RBCs, person does not have D antigen, it means that person is Rh negative. If you inject him D antigen or D positive RBCs, person might produce a strong reaction against D antigen. Person may produce anti-D antibodies. Person may produce anti-D antibodies. But other antigens are not so strongly immunogenic. They are immunogenic, but they are not so strongly immunogenic as D antigen. Right? And if D antigen is the most important and most strong antigen and most immunogenic, so in common medical practice, when we talk about that person is Rh positive, actually we are talking about that person is Rh positive person is equal to D positive. In common and general medical practice, when we say that my patient is Rh positive, actually I mean person is D positive. And if I say the person is Rh negative, it means person is D negative. So now onward, in our discussion during the lecture, we will follow this simple rule that when I say someone is Rh positive, if I don't mention specifically about C and E, it is understood I'm talking about D. So D positive people are Rh positive and D negative people are Rh negative. Is that clear? Any question up to this? Now, few more words about D antigen. Okay, let me make red blood cells, and these are 3 red blood cell, okay, 4 red blood cells. A very important terminology. Let's suppose this is the D antigen. Okay, I will make it more simple. This D antigen I'm just showing by D. Now, these are D antigens present on this red blood cells. So of course this will be D positive. It will be D positive red blood cell. There are no D antigen. If there are no D antigens, what is this? D negative. Is that clear? So this is what an average doctor should know very well. If average doctor doesn't know this, it's very sad situation. The minimum a doctor should know about Rh system is, if person is having D antigen, person is Rh positive, say loudly. And if person does not have D antigen, person is Rh negative, right? But now move on. Some people have D antigen but very few molecules. Very few molecules. The density of D antigen is very less on red blood cell. If density of D molecule is very, very less, we call it weak D. What we call it? Weak D. So we have Rh positive patients, or people who have Rh negative, or D positive and D negative. And sometimes we come across people who are weak D. Why we are talking about weak D? Because sometimes weak D people have very less D molecules, right? And they may not be sometimes detected by the some of the available antisera. So weak D means that, okay, let's suppose we just call it D antigen is mean dog. We're just talking about it. So there's so many dogs. There are so many dogs, and D positive, D negative, no dogs. And weak D, few dogs, few dogs. Now, every dog has how many legs? 4. D antigen also has 4 antigenic determinants. D antigen has 4 antigenic determinants or 4 epitopes, right? Now, here are 4. Okay, let me put it they are having 4 upside down dogs. No, they are not 5. Okay, let me make their more artistically. Let me try my art. These are the dogs. These are D antigens, right? 1, 2, 3, 4. 1, 2, 3, 4. So they are having 4 legs. These are normal D antigen with high density, and these people are called D positive or Rh positive. These people don't have these dogs. If they don't have these dogs, these are D negative or Rh negative. Very good. Now here there are very few dogs. Very few dogs. But dogs are normal but very few. Right? So this is called weak dog. Then another category is partial D. Partial D. Now what is partial D? The difference between partial D and weak D is that some of the legs are broken. 1 or 2 legs are missing. D molecules are there, but maybe only 2 legs, or only 1 leg, or only 3 legs. So it means as compared to D positive, weak D has quantitative difference. Quantitative difference. And as compared to the D, partial D has qualitative difference. Quality of the D antigen is not right. Right? So I'm just introducing the few terms. First of all, D positive people who have D antigen. D negative people who do not have D antigen. Clear? This is for a good doctor. A very good doctor, sometimes we come across weak D, where D antigens are normal but less in number. Or sometimes we come across patients who are partial D. Partial D are those people who are having slightly abnormal, qualitatively abnormal D antigens, or missing some of the epitopes. Is that clear? Okay. Then another term is, we talked about D positive, D negative, weak D, partial D. Another is Rh-null. Another term is Rh-null. It's rare, but what is it? Yes. Yes. Rh-null mean? There is no dog. No dog. But D negative is also having no dogs?
But there is also no partial.
No. Listen, my friend. Here D antigen is present. Here D antigen is not present. Here D antigen concentration is very low. Here D antigen, whatever concentration, is abnormal structure. Then another term sometimes we come across in the literature, Rh-null. What does it mean? If we say there is no D, then it should be called simply D negative. So there is something more.
So there is neither D nor E or C.
Excellent, very good. Actually, Rh-null is a person who is considered no D, no E, no C. None of the Rh antigen is there out of important, because, come here, when we say D positive, we are talking about D, but we are not talking about C and E, which may be present here. In the same way, when we say D negative, right, it means even though we say generally speaking Rh negative, but there may be C or E or may not be. You are understanding? But when we say Rh-null, it means there is no D, there is no C, there is no E. Now listen carefully, come back. I said there are 2 genes. I said there are 2 genes. Now in this person, Rh-D gene is expressing very well. Here Rh-D gene is absent or totally non-functional. Is that right? In this case, Rh-D gene is either under-expressing, or molecules which are produced cannot be expressed on the membrane, or their gene is mutant in such a way that some of the products are abnormal, right? But in all these stories, we didn't talk about Rh-CE, right? When actually there are no products from here as well as no antigenic product from here, then we say this is Rh-null. These 4 terms are clear?
Clear.
Any question up to this? Okay. Now let's move forward. Now we know if a person is having D antigen, we call him Rh positive. Or we simply call that positive blood group. Or we simply call it positive blood group. And a person who does not have D antigen, we call him negative blood group. We call him negative blood group. Now we come to the how many blood groups we can have. As we know that, let me draw red blood cells here and draw the RBCs. Okay, these are the red blood cells. Now let's start with simple thing. First of all, we classify these red blood cells according to ABO group. Let's suppose it has sugar oligosaccharide A. What is this blood group? A, right? This has only B antigen. So what is this blood group? B. If it has A and B both, what is blood group? Yes, AB, very good. It doesn't have A or B. It has only actually H antigen, no A, no B. Because remember, A and B antigens are basically modified H antigen. So when H antigens are there on red blood cell, but they are not modified by addition of terminal monosaccharide, they are not modified into A or B, we simply call them, they don't have A, they don't have B. But it doesn't mean they don't have any antigen. They have H antigen. Is that right? So, but those people's red blood cell which do not have A and they do not have B, we call them traditionally which blood group? O. Is that right? Am I clear? And so these were about the candies. If you have candy A, blood group A, but with the candy, if you have candy B, blood group B, if you have 2 types of candies, so sweet like me, blood group A and B, and no candies, this is O. Is it clear? Now you imagine, with the candy you put the chicken piece, D antigen. This is D antigen. So what we'll say, this according to ABO system it is blood group A. According to the Rh system it is positive. So it will become A positive blood group. If you put a chicken piece here also, this will become B positive red blood cell. Right? If we put a chicken piece here, I mean D antigen, then what we say? Person will be A, B and positive blood group, AB positive. And if a person has D over here, chicken piece is there but no candy. But there is no candy, only wrappers of the candies are there. There is no candy, then what we call it? O positive. It means if someone has red blood cell with only A antigen, it is A and no chicken piece, it is A negative. If someone has someone has B antigen but no chicken piece, so what we call it? B negative. If someone has A and B candies, yes, oligosaccharides, right, then we'll say blood group A and B but no D, so we'll call it AB negative. And someone who has only H antigens, no A, no B and no D, we'll call it O negative. So this is how we can classify red blood cells by 2 systems, ABO system and Rh system. Is it clear? Actually there are so many blood groups, more than 35 systems of blood groups, but 2 are most important, ABO system and Rh system, right? So here I have shown ABO system and Rh system. One more thing which I want to make it clear is that in the beginning I said that people who are D positive, they are also considered Rh positive. What is the distribution of Rh positive and negative in the population. Actually, in American whites, about 85% people are Rh positive. They are Rh positive. But you go for American blacks, then it is about 95 American blacks are Rh positive. But if you go for African blacks, 100% of the African blacks are Rh positive. And if you come to Asians, almost 99% are Rh positive. So we can say, when we say Rh positive, it means D positive. You can see that D antigen is very, very common in 85% of white Americans, in 95% of black Americans, 100% of African blacks, and Asians, 99% population has D antigens. It means in the world population, majority is D positive or Rh positive, right? Now, after this, we'll discuss other aspects about these antigens, right? First, we'll have a break, and then after that, I will continue with some important clinical differences between the Rh antigens and ABO antigens.